Discovery of viruses cannot be attributed to a single person, but it has been a joint contribution of a team of scientists, including Dutch, German, and Russian microbiologists. It was the Russian microbiologist Ivanovsky who in 1892 used the filter devised by Charles Chamberlain in 1866 to trap smallest possible bacteria that were supposed to cause the disease in the tobacco plants called as tobacco mosaic disease. It was Bijernich from 1851 to 1932 who deserves the credit of first realizing that the causative agents of tobacco mosaic disease differ considerably from microorganisms. And Stanley in 1935 first crystallized the viruses and proved that viruses have both living and non-living attributes, which opened a debate on their nature, whether they are living or non-living or on the borderline of the two. Bacteriophages, that is the viruses which infect bacteria, were first discovered by a French scientist de Harel in 1917, while Schlesinger in 1933 first determined the chemical composition of a virus. Hershey and Chase in 1952 were the first to demonstrate that the genetic information is carried in the phage DNA and they have to infect the host cells for transmission of this information. The important and pioneering contribution by these microbiologists guided the way forward for the recent advances in the field of virology. Evolution of viruses is still an intriguing question, and three hypotheses have been put forward to explain their origin and evolution. They include: first, viruses, because of simplicity of their genome, might represent the direct descendants of earlier self-replicating units in the primordial soup from which the first cells evolved and then co-evolved to give rise to the complex life forms. Second, viruses evolved from free-living organisms that invaded other life forms and gradually lost their function. This is also known as retrogressive theory of evolution. Third, viruses are escaped nucleic acids no longer under the control of their host cells. This is also called as the escape gene theory of viral evolution. While all these theories or hypotheses partly contribute to the understanding of the viral evolution, a unifying concept to explain the same is yet to emerge. All viruses have two basic structural components: the core of nucleic acids, what comprises their genome, and a capsid, that is the shell-like covering around this genome. The two together, genome plus capsid, comprise the genocapsid. So far the genome of viruses is concerned, they are too flexible in this and employ all four possible types, including single-stranded DNA, double-stranded DNA, single-stranded RNA, or double-stranded RNA. Single-stranded DNA for example parvoviruses, double-stranded DNA, for example lambdaphages, single-stranded RNA, as is found in tobacco mosaic virus or Rome mosaic virus, and double-stranded RNA as in retroviruses. Capsid is a coat of nucleic acids surrounding the genome, which is composed of capsomers, the protein subunits, the number of which, as I told earlier also, is species specific and an important taxonomic criteria. Some viruses do have an envelope, lipoproteinaceous covering around the capsid, what are called as enveloped viruses. Viruses can be broadly classified into three morphological types, including icosahedral, helical, and the complex. Icosahedral, or a typical icosahedron, is a polyhedron with 12 vertices in 20 equilateral triangles as faces. The capsomers in the icosahedron viruses are composed of protomers, which may be either pentamers, that means with 5 protein subunits, or hexamers with 6 protein subunits. The best examples of icosahedral viruses include adenoviruses, polioviruses, and canine parvoviruses. Helical viruses are basically the rod-shaped, long, hollow cylinders with linear array of nucleic acids and protein subunits making up the capsid. In such viruses, for example tobacco mosaic virus, the protein subunits are arranged in a helical spiral with genome fitted into a groove on the inner portion of the protein. The size of the helical capsid is influenced both by its protomers and the nucleic acids enclosed within the capsid. So for the complex types of viruses are concerned, these are the viruses that are assembled from the parts that are synthesized separately. That means head, tail, or the capsomers come from the different parts and they do not fit either into the helical or into the icosahedral symmetry, and as such are called as complex in their morphology. And bacteriophages or pox viruses are the best examples of complex viruses. An interesting thing to know is how do viruses replicate? Viral replication differs in different types of viruses, but the broad sequence of events in different types of viruses are almost the same. The 5 principal stages involved in the process of viral replication include adsorption, penetration, synthesis of viral nucleic acids and the proteins, and assembly or maturation of the virion particles, and finally their release. So for the attachment or the adsorption is concerned, this is the first step in the viral replication, which generally takes place through random collision of virion particles with the host cell surfaces, facilitated by the specific receptor proteins on the host cell membrane or even attached by the ionic interactions. Then the penetration of viruses so far is concerned, it refers to the entry of viruses into the host cell followed by their uncoating. That means the removal of the protein coat surrounding them through the activity of proteolytic enzymes. After their penetration into the host cell, the next step is replication or the multiplication of the viral genome. And different viruses have different strategies for the multiplication of this genome. Once abundant viral genome and the proteins and the other components of their capsids are synthesized within the host cell, the important step is their assembly, which is the 4th important step in their replication. And after they are assembled within the host cell, finally the step is the release. That is, viruses, mature virion particles have to get released from the host cells